Hi, good morning. I'm Alex Zuntini. I'm a research fellow at Royal Botanic Gardens Kew, where I've been working for 2.5 years with Angiosperm's phylogenomics, and today I'll be talking about target sequence capture on Museomics. I'll be talking initially about what is target sequence capture, then how it works, and then some case studies in plants and how we can use this knowledge to improve the protocols. But before talking about target capture, we need to understand why we do it and why is it good for herbarium plants. So some 15 years ago, when I was doing my PhD, I was doing phylogenetics in Bignoniaceae, a family of Lamiales, and we were using two markers from plastid, NAJDF when one spacer, and also one nuclear marker, the Pepsi gene. And as you can see here, this black lines represents the amplicons that we use to do Sanger. And as you can see, the size of these amplicons are quite big, like 1300 base pairs, 1100 base pairs. And well, it simply didn't work with herbarium material. So what I had to do was, okay, I want to use old or really old specimen like this. There's nearly 200 centuries old. So I had to design new primers, so I could focus on smaller amplicons with a smaller overlapping as well, which made me, instead of doing just 2 PCRs to amplify one marker, you need up to 5, sometimes 6 PCRs to complete the marker, and it was quite a big work. And to do all of the 3 markers, it was a big work. And still, as you can see, the amplicon size was still quite big, like all of them above 400 base pairs, which for a berry material, it can be a long shot, especially for really old specimens. So what's the alternative? Well, the alternative is doing genome fishing. We call it target sequence capture, but it can also be known as bait hybridization, target enrichment, HybSeq, sequence enrichment. There's many names for it, but the basic idea is we get our pool of DNA and we just gonna fish the genes that we are interested in. So how target sequence capture works? You have your fragmented DNA with your target sequences, here marked in blue, and your off-target sequences, those that you're not interested in, marked in orange. The target sequence can be anything, can be genes under neutral selection that you may use for phylogenomics. It can be spacers or no-coding regions that you might want to do population genetics. It could be genes related to functional traits. You pick, depending on the question, you target whatever you want. But the protocol is basically the same. You're just gonna denature the DNA and the baits, add the baits, and they will hybridize with your sequences. So for example, here the baits in this light blue, it will bind to the target sequences. Not all of them, sometimes you miss because it's a random process. And these baits, they are biotinylated. So they will, when you add magnetic baits with streptavidin, the baits will attract the single strand of DNA that it's hybridized. You can use a magnet to separate these beads, wash out everything that you didn't want, and then you amplify this, and you end up with what we call enriched library. This enriched, we say enriched because it's not filtering, meaning there will be things that you don't want. For example, we can see here an off-target sequence, but also you would have what we call a splash zone, which is the fragments that are between. When you have a fragmented DNA, you have your target sequence, but you have the flanking regions of those, which could be an intron, could be a spacer. So in this approach, you can recover ideally your target sequences. Sometimes you can recover off targets, and in case of plants, it could be ribosomal DNA, it could be plastids, mitochondrial DNA, it could be repetitive elements. Those that are in higher dosage are more likely to be detected. But you can also have the splash zones, and as I say, this could be introns or spacers, which, depending on the kind of work you're doing, phylogenomics, population genetics, will be much more variable than a coding region, and therefore may have much more information available for you and more phylogenetic signal, for example. A really interesting thing from target sequence capture is that it allows working on non-model organisms, because although you have baits, you don't have primers, which are usually much more specific. Baits are longer than primers, so you can develop, for example, bait for a whole family, and even if you don't know every single genus, you don't need to have sequence of all the genera in a given family for the bait set to work. And most especially, it allows using degraded material, because we start with fragmented DNA. But it's not any kind of fragmented DNA. We must start with short libraries. Okay, so if I must start with short libraries, what is the process? How do I go from the DNA to sequencing? Well, if it's DNA, it starts with an extraction and followed by cleaning or not, depending on the group you're working. You may want to do a silica column or bead purification if you're working with families with latex, for example, or really nasty secondary metabolites. Despite the way you do the extraction, the next step will be profiling the DNA, meaning let's assess how degraded it is from very low quality DNA, super degraded, to somewhat degraded, ranging from high to low molecular weight, to a high quality DNA where most of it is high molecular weight. And also we want to measure the amount of DNA, the concentration of DNA, so we can start the protocol with even quantities so the process becomes more predictable. And as I said before, the target sequence capture uses fragmented DNA. So the next step here will be fragmenting it. Probably the most common way to fragment the DNA in the lab or shear the DNA in the lab is by sonication. Sonication is a process where you submit your samples to a sound treatment in water by submitting the samples to a high intensity ultrasonic sound that, by the intensity of it, it creates cavitation bubbles due to the negative pressures, and the implosion from these bubbles results in the shearing force that breaks the DNA. And as you can see in here, if you start with a high quality DNA, the longer you submit the sample, the shorter will be the average size of it. So you can see that in the beginning it's quite easy to break it, but it tends to get harder and harder to really get rid of DNA. So what we do, since we know what is the curve and we know where we want to produce a library, for example, at this range, if we start with a high quality DNA, we know we need, let's say, if it's 10 seconds each, we can say 1, 2, 3, 4, 5. 50 seconds would bring to this 60, 70. And it's good to do this kind of profiling, because if you have a sample that looks like this and you want to reach this, you just need 30 seconds. But this is not the only way of shearing DNA. There's another way, which I like to call hybridization. And it's basically collecting the plant, put it in a press, mounting in paper, put in a cabinet, and leave there for 10, 20, 30, 40, hundreds of years. And it will be sonicated. Not sonicated, but degraded. And this is probably the most exciting thing about target enrichment, because the herbarium is doing part of the job for us. Sometimes it does too well and fragments too much, which can be troublesome. And it's quite costly if you think that you must maintain a building for years and years. But jokes apart, it means that since part of the process is fragmenting the DNA, we can use all these vast collections that we have in our hands and exploit all the genetic potential in there. So after we shear the DNA, depending on the degradation profile of it, we start doing the library prep. So we can't simply bind the DNA to anything. It must be sequenceable. And to be sequenceable, we must get our inserts of DNA, or the fragments that we are interested in. We put an adapter, here marked in yellow and green. Another thing we can do is add some specific barcodes or indices. We can see here in red and blue, in blue as well. And it allows us to sequence more than one sample, because this will be unique for your sample. So you can mix many samples together, and in the end, after you sequence, you just check what are the boundaries of the sequence, and you can say, by the combination of this is either sample A, B, or C. And this process is quite simple. It takes some time, obviously. It involves PCR, but it can be done with any kind of DNA. And the first step is end repair. So if you have a degraded DNA, you will replicate the DNA to have a blunt end, which allows for the adapter to ligate. And since this library prep involves PCR, if you have herbarium materials that you know the concentration is low, you can work on the PCR and improve the amount of library you get in the end by just doing more cycles, as we did with Sango before. So after we do the library, we're going to pool the samples, meaning we're going to put libraries together to be hybridized. And we can only do that because we use those barcodes or indices I mentioned before. So we can sequence many things together and then bioinformatically separate them later. And once we do the pools, we go for the hybridization as I described before. And once that is done, we go for quality control, see how everything is. We prepare samples for sequencing. We get the sequencing results. We sort them by sample by using the adapters, the barcodes, and then we go for data analysis. Data analysis is an entirely universe. I'm not discussing it today, but there are ways that you can improve the results by tweaking your data analysis pipeline. But this is something that you can do as you learn, whereas all the other tricks that I'll be bring in here are things that must be done in the lab. So once you do the libraries, it's done. You can try it again. Whereas a pipeline, you can just spend a bit more time working and learning. So first thing is, does it really work? Yes, it does. Here there is Melissa begonifolia, a 200-old specimen of boxwood collected in Santa Elena that we did libraries, and you can see on top here that the genomic size distribution, and in the bottom plot the library that we managed to produce from it. You can see the size is nearly the same, but it's important to bear in mind that when we say library size, we're not saying all of it is the target DNA, because you have the adapters, as I said before, that are part of this. So if you have 300 base pairs, it means that you have roughly half of it will be the insert size with your target insert. Okay, so if it works for Melissa, how does it work for everything or anything? Can we just target all angiosperms? Yes, it's possible, and it was done by Matthew Johnson and allies, who designed a bait kit that targets 353 nucleogenes across angiosperms, and it works really well. And they did it by getting transcriptomes across angiosperms from the 1KP project, sequencing, selecting the genes that were orthologs, and then developing the baits, here marked in blue, orange, and pink. They cleaned this, and in the end, let's say you have this species that wasn't part of the kit, that you can use the similarity of the beads to recover these targets and end up with here the sequence in black for the target regions, but also the splash zones, as I mentioned before, which are those that are not entirely covered, targeted by the probes, but that still can be recovered. This kit is made of like more than 80,000 probes. Sometimes 5 to 15 sequences were used per locus, and it targets up to 260,000 base pairs, and the average recovery is half of that. So a couple years ago, we wanted to assess, okay, how well is this kit performing? So we used the initial data set we have in hand, something like 800 samples, and we tried to understand what are the factors affecting target sequence capture on this bait kit on herbarium materials. So the first assessment of the performance of target capture was to understand how the quality of DNA was affected by the source and the age of materials. So one totally expected result was that older specimens tend to yield low quality DNA, and more recent ones higher quality. No surprise there. Also, silica materials will more likely preserve better the DNA and lead to higher quality DNA. When we look at the habitat of where the species occurs, we don't see any striking pattern. But when we correlate that to the way the material was collected, then we see a pattern that, for example, tropical plants in silica, they don't preserve so well as desert dry plants. So first we could see that the DNA quality can be affected, so by age and storage and the habitat. The next step was to evaluate the actual gene recovery. So what we did, we used HiPiper, which is a standard pipeline to recover the genes, and we plotted it by age and those three categories again, and we can see that the genomic concentration of DNA falls through time, that is expected because the DNA, as it gets degraded, it's harder to extract and you end up with less DNA available in the sample. But interestingly, although the concentration of genomic DNA is falling, when you compare the genes retrieved above 50% of the target, which means like if you had a gene that is 1,000 base pairs, you consider you have it if you have 500 base pairs of it. When we look at the profile of silica dry samples in blue and recent herbarium materials, the pattern is quite the same. It's a bit more, there's a longer skew on the herbarium materials, but the results are still quite satisfactory. Whereas for old specimens, the concentration of DNA falls, but you end up with much more variation. So it's much more unpredictable if you can have good results from herbarium samples. Another thing that we wanted to investigate was about the taxonomic bias that could be affecting the recovery of samples. And it was really interesting to see that when we break those samples by the top 10 families in the data set we had, you can see that genomic concentration sometimes varies a lot within a group. Sometimes it's quite stable, it doesn't yield much. For example, Cyperaceae is really hard to get lots of DNA, whereas Sapindaceae, sometimes you get like tons of it. Poaceae, wow, it's amazing. It's not in this data set, but you can get a lot. The enrichment efficiency, which is the number of reads that are in your targets divided by those that are sequenced, it also fluctuates, and that's basically how much of the DNA you sequence is really what you wanted. And it is related to taxonomy, obviously, because the closer you are to the target phylogenetically, or not the target, the model for the sequence, the higher the efficiency. But it's interesting to see that although the efficiency varied a lot, you could see that genes retrieve above 50%. It varies quite significantly in some groups. For example, in Melastomataceae, Myrtaceae, Rubiaceae, older herbarium materials compared to silica materials, the performance is really, really contrasting. Whereas in some cases, for example, Cyperaceae, Chrysobalanaceae, Sapindaceae, it really doesn't matter the age of the specimen. It performs just well. And we believe it might be related to the traits that these groups they have, or it could also be related to the proximity between the groups and the sequences used to develop the target. This is still to be further investigated. Since we saw that habitat and storage were related to alterations in the quality of the DNA, we thought it could also alter the recovery of the genes. So when we look at genomic concentration across groups, we don't see like a major difference between the materials where they were stored and the climate, but we do see variation in the enrichment efficiency, remembering number proportion between the reads that are mapped, that are targeted over everything that was sequenced. And we can see the most contrasting pattern observed in wet tropical areas, where old materials are much less efficiently hybridized than silica materials. And in the end, it translates into a worse gene recovery. So when we ask what is the proportion of genes recovered of at least 50% of the target length, we can see that in the wet tropics, silica, they usually get a lot or most of the genes, and older herbarium specimens, they underperform so much, whereas either drier or colder areas, they tend to perform better despite the time. And we believe these changes in the climate and in the habitat might be related to traits of the plants in these different areas. So with this small data set of nearly 800 samples, we could learn a bit about how habitat or taxonomy can affect the recovery and learn from it and improve the methods as we were doing. But 2.5 years passed, and now we have much more data, more than 5,000 libraries done, and we want to understand more about the data set to make the next steps easier. The first thing to look is how gene recovery is related to genomic DNA concentration. We can see in here the sample concentration in the log scale compared to the total length of recovery, meaning summing the length of all the genes recovered. We can see that it's more dense up here, so the higher the concentration, more likely we get longer sequences. We can see a bit bias here because the libraries are also longer, because there is usually some correlation between concentration and size of the library. We can see that it does get a bit more, and shorter libraries, they tend to underperform, but if concentration is higher, you give it a bit more chance. And why is that? Because when you have a higher concentration of DNA, it means you have a higher diversity of the DNA that will lead to a higher diversity of libraries, and once you go through hybridization, a higher diversity of reads that are sequenced. And you need different reads to assemble a whole gene. If your DNA concentration is low and you need to do a lot of PCR cycles to reach the minimum amount to sequence, you are just sequencing the same thing more times, so you don't have a good diversity. So the lesson here is, if your DNA concentration is low, before doing any library preparation, try to concentrate it. If you did an extraction in 100 microliters, dry it to 25, for example, or if you still have material left, do 2, 3, 4 extractions, combine the DNA from those, concentrate it, and use all of it to do libraries. It is much likely to lead to a better library. The next thing is to look at how the library size is affecting. I mentioned there was a bias, and we can see it here when we compare the number of genes and the total length. We can see that as you get more genes, you start obviously to get longer, but if you look at any point, either like in the y-axis or x-axis, you can see that shorter libraries in green, they either take more genes to get to a same given length, or the same number of genes will lead to shorter sequences. So longer libraries can yield more genes, and longer libraries will yield longer sequences. And most importantly, longer libraries will lead to longer splashing zones, which are those that are target. And why longer means better in this case? Because we have more potential sites for the baits to work. So if you have a sequence that is dissimilar from the baits, if you have a longer insert size, it means there are more potential sites for any bait to bind. And this is really important because when we are talking about baits, we are talking about hybridization. And the step immediately before hybridization is the library pool. So when we talk about performance of the size of the library a really important factor is the pool. So let's look into the pools in detail. Here I selected some nearly 100 pools. Each column is one pool. Again, we have on y-axis the total length of the recovery, and the colors in each hybridization pool are green short libraries going through warmer colors the longer the library is. And we can usually see the pattern that when you have libraries that, for example, when you have a pool with a great variation, you will see greens in the bottom, warm in the top, warmer colors, green colors, warmer colors, and green colors. So this is the kind of pattern. Just to look at a bit further, see some examples. Each of these plots is a selection of those pools I showed before, with good examples in the top row and bad examples in the bottom row. I call them good examples because you can see that the libraries, they have the same size, and the size is here in the x-axis. So they are clustered in the x-axis, leading to a good recovery, here measured as the total length. And although you may see underperformance in groups, we can see different families in each of these plots, there is still not much variation within a pool. On the bad examples, we can see that if we mix a higher range of library sizes, the shorter ones will underperform and the longer ones are winning. And we can see this pattern, like even changing the families, the mixing short libraries with long ones will usually lead to another performance of these. So more homogeneous, less discrepancy. And why is that? It's because during hybridization, the libraries are competing for the baits. Let me explain a bit better. You probably remember when I explained before, library size is made of the insert size, which is your actual DNA, and you have the adapters on the side. So let's say you have a bait of 120 base pairs, which is the angiosperm 353 size. The first step is to block the adapter, and then we're going to denaturate these libraries and add the baits. These three hypothetical samples, A, B, and C, they have different library sizes, and it's not the adapters that are varying, but the inserts, right? Sample A has a longer insert, so by chance, more baits can bind to it. Sample B is a bit shorter, and you can see that not a whole bait, or nearly a whole bait, is binding to it, whereas sample C is much shorter than sample A, and even like probably less than half of the bait is binding. So when we think about free energy in the system, sample A is more likely to be kept through the process because there will be more bonds between the bait and the target. So sample A, the chances that the bait will bind to sample C for any given gene is much lower than it will bind to sample A. So in this fight, sample C loses. And it's interesting to see that even when we have really short libraries, if there isn't much variation in size of the library, if the range is small, you can get really good results. There will be cases where it's underperforming and it could be due to excessive PCR to reach the amount of required libraries to do the process. There can be many other things affecting it. But if these were sampled with longer libraries, this will be much lower. So what can we do then? Well, since we know the baits are binding in a temperature control environment, the first thing you can do is lower the temperature, because it's just like primers in a Sanger sequencing, in a PCR, sorry. If you lower the temperature, less stringent will be the reaction, allowing the primer binding more easily to the similar sequences, or in this case, shorter. We can do shorter baits. So for example, if we have a bait that was 120 base pair and it's not working for your group because you have mostly degraded DNA, you can develop baits for 40 base pairs. And this was an approach that was done in the beginning of target sequencing to do population genetics of woolly mammoth. They developed a target capture for mitochondrial DNA of mammoth, and they managed to get amazing work because the baits were really short, and it allowed them to capture mitochondrial DNA up to 5,000 years old. So you can develop shorter baits. You can also increase the overlap of the baits. In here you can see that they overlap by maybe a third. They can overlap for more than half if you think the samples are too degraded. Obviously, it will lead to a higher cost, so you have to decide exactly what you can afford. And another way is developing specific bait kits. If you go to many journals nowadays, like Application in Plant Sciences, Molecular Ecology, Resources, Molecular Phylogenetics and Evolution, you'll probably see in every single volume a new kit for one family. And sometimes you have competing kits for the same family. So it's out there, and there are many options. So how all of this works for angiosperms? Well, all of what I presented was developed as part of the PAFTO, which is the Plant and Fungal Tree of Life project in Kew Gardens, which are aiming to produce phylogenetic frameworks of all genera, sampling one species per genus, in plants that represents 13.5 genera. And one could ask, does it really work? Well, I hope I proved with the data that I presented already, but let's say you don't believe it. You can take a look on 2 special issues that were published earlier this year in American Journal of Botany and Application in Plant Sciences, just evaluating the Angiosperm 353 kit in different data sets, different groups, taxonomic levels, from ordinal phylogenies to closely related species, even population genetics sometimes. We can also see some works using the off-target to combine target capture with Sanger data. There are many approaches that are being developed with this kit, and also not only Angiosperm 353, but you're gonna see this myriad of kits being developed, and all of this is unlocking all these resources for us. And how does it work for angiosperms? Well, from herbarium, I can say it works really well. We are currently working on a big phylogeny for more than 8,000 genera using nuclear data captured with Angiosperm 353. And I think somewhere around 5,000 of this came from herbarium specimens, and it allows us to sample all the families and all orders in angiosperms. Not only that, it also shows that it works across everything with a really good representation of all lineages. So what we can say is, well, target sequence capture really works for material from herbarium. It usually underperforms. Yes, this is expected when you compare to recent materials, but still, the herbarium represents a highly valuable source of genetic information, especially for groups that are hard to collect, things that are extinct, or they're in regions with conflicts, or that you want to get information from type collections. It's such an amazing opportunity to be able to address those, and target sequence capture is currently the only way of doing that. The performance of the kit, of the hybridization, can be affected by not only time, but taxonomy, the habitat, the way the sample was preserved. But if we are careful enough, we can use that information to inform how we're going to sample our group. If you give preference for not only an older material, but from a drier environment, or if you are designing a study where the specific group is not relevant, but more the region, you could give preference for a group that you know that tends to perform better from dried material. And once you do all of that, you can still adjust the protocols to do the library and the hybridization and get much better results from it. This was all I wanted to present to you today, and all this work was only possible because of this huge team, the pivotal team at Q, with amazing people. This work was funded by Kaleva Foundation and the Sackler Trust, and also super well sponsored by Q. And most importantly, it's only possible because not only we had an amazing team at Q, but we have a network of more than 300 scientists around the world helping us to deliver all of that. So with this, I would like to also thank the organizers for inviting me to present here part of my work and share with you some insights on how we can do target sequence capture in plants. And I'll be really happy to address any questions you may have now, or in the future you can connect it to me via Twitter or my email account. Thank you.